Hello, I'm just going to do a quick revision video on cell division, stem cells, so use of stem cells in plants and animals, and then looking at cell division, so mainly mitosis. So the key things to focus on in this video are going to be mitosis and the cell cycle, cell differentiation, specialization, what they are, and of course some examples of specialized cells and what makes them special. But predominantly we're going to focus on stem cells, so the function of stem cell, definition. We're going to evaluate the many uses of stem cells, so adult, embryonic, and then a little bit of a focus on plant stem cells as well. So before we can look at stem cells, we need to focus on two key words. So cell specialization and cell differentiation. So cell specialization does exactly what it says on the tin. It's when a cell becomes specialized at a particular job. So it's when it gets a particular function. And a cell becomes specialized through a process of differentiation. Which again does exactly as the word says. It's when a cell becomes different. In animal cells, differentiation happens at an early stage. So during developmental area, differentiation occurs where the cells become different, so they become specialized cells, and then the process kind of later in life is just for growth, so for repair. In plants, they maintain the ability to go through the process of differentiation at all stages. I'm going to focus on particular parts of a plant that allow that to happen in a second. So this page then looks at the main examples of specialised cells that you're going to come up with in the exam, so animal and plant cells. What you have to think about, whenever thinking about specialised cells, you have to think about the cell, the function of the cell, and then what is it about that cell that has made it good at its job. So how is the cell adapted to its function? So the example, sperm cells. So sperm cells contain, their function is to carry half the genes during fertilization. It needs to be able to move quickly, so it has mitochondria, is probably the main adaptation of a sperm cell because it produces energy which allows it to move quickly. A red blood cell, its main job is to carry oxygen, its only job is to carry oxygen. It has a large surface area, which means it can contain more hemoglobin, which means it can transport more oxygen. A nerve cell also has a large surface area, so it can make contact with more cells, so it can transmit more messages. A palisade cell has a large surface area, so it can absorb more light. A root hair cell has a large surface area, so it can take up more mineral ions from the soil. A ciliated epithelial cell has a large surface area. It also has cilia, which their function is to move things, so particularly find them in the trachea, where they move mucus out of the body, and you also find them in the fallopian tubes or the oviduct, where they move the egg from the ovaries to the uterus. Then you have a muscle cell, which again, the muscle cell's function is to produce lots of energy. So its primary adaptation is to contain lots and lots of mitochondria to produce lots of energy. But you will see, if ever you're in doubt as to what adaptation to give as a cell in terms of its function, just go with surface area, because the vast majority of them are adapted for surface area in particular. I'm talking about exchange. So now we're going to focus on mitosis. So mitosis is the production of a new cell. So it's how we produce new cells from cells that already exist. So I have a cell here. So we start with a cell. Inside the cell, of course, in a eukaryote cell, we have a nucleus, and inside my nucleus, I contain my genetic material. Okay, so in this cell, I've just drawn it mainly to save time. I've just drawn a cell that contains two chromosomes. Okay? Rather than saying 2 chromosomes, I could also say it contains 1 pair of chromosomes. Question could come up, why do we talk about chromosomes being in pairs? One comes from the maternal side, which is your mother, and one comes from the paternal side, which is the father. If we were talking about a human cell, then of course this cell would have 23 pairs, but I'm not going to draw 23 pairs. So we start off with chromosomes like that. This example's got 2 pairs. The very first stage of the cell cycle and mitosis is DNA replication. So that is the first stage. And what effectively happens is my chromosomes now look like that. Okay? I still actually only have 2 chromosomes. I still actually only have 1 pair of chromosomes, but each chromosome has replicated itself. And then all that happens is this splits into to produce 2 cells. To do better than that, we can say it produces 2 genetically identical cells. We could also use this word, it produces clones, which are genetically identical cells. So this cell is exactly the same as this cell, which is also exactly the same as the original cell that it came from. And this is an incredibly important process. It's used in growth and repair. Most people think it only happens in kind of bacterial cells. It actually doesn't happen in bacterial cells at all, because bacteria divide by binary fission, not mitosis. This is how all cells grow and how all cells repair. So it's happening in us all the time. It also happened in the very early stage of development. So you have a sperm cell and you have an egg cell. They come together to produce this thing here called a zygote during fertilization. And when that zygote divides to form a ball of cells, which is called an embryo, that is the first stage of growth in your development, which is mitosis. So mitosis is the growth and development, growth and repair of cells. But whenever you damage something, you also need to repair it, and that also happens by mitosis. The main focus then of this video is going to be looking at stem cells. So stem cell is an undifferentiated cell. So if you look at that word again, undifferentiated, which means it is not different. So it's an undifferentiated cell that can differentiate into any cell or organ. So again, in that embryonic stage, embryos are an example of a stem cell. So in the embryonic stage, these embryos are undifferentiated, which means they do not have a job. But eventually, due to stimulation by hormones and other chemicals in your body, they get told to become something. So this one might be told to become a red blood cell. So they differentiate into a red blood cell, which is an example of a specialised cell. This one might be told to become a nerve cell. This one might be told to become a skin cell. This one might be told to become a muscle cell. So stem cells are undifferentiated cells that can differentiate into becoming any cell or organ. There are 3 main examples we need to be aware of. One is animal or human embryo cells, which I said can become any cell or organ. We also then have adult stem cells which exist. So in your body now, you have something called adult stem cells. In particular, they exist in your bone marrow. and they can differentiate into a group of cells or organs that we'll talk about in a second. And we're going to talk about plant stem cells. So we're going to focus on human stem cells. And human stem cells are phenomenal things. Okay, they have the ability, as I said, they're undifferentiated cells. They can become any cell or organ, and they've got the potential to cure a whole number of diseases. So they can cure diseases. And I'm just going to talk about a few specific examples that you need to be aware of. So one, say for instance somebody needed an organ replacement. What you can do is you could use stem cells to create an organ. So imagine somebody was a diabetic and their pancreas did not work. We could use stem cells to create them a new pancreas. Say for instance somebody was paralysed, which means their spinal cord has been severed and they can no longer walk, and some people obviously can't walk or actually even use their arms. We could then use stem cells to regrow part of their spinal cord, which would cure paralysis. In order to do either of those two things, you would have to use embryonic stem cells. You could, though, use adult stem cells as well. Adult stem cells are far less invasive. They don't have the potential for life, so some people do prefer to use them. They've got less ethical issues. So an adult stem cell, as I said earlier, adult stem cells are found in your bone marrow. Every single blood cell, so red blood cells, white blood cells, all different types of platelets, all different types of white blood cells exist, all come from your bone marrow. So you can use adult stem cells to make any type of blood cell. Plant stem cells then, I'm going to give them their proper name now. We call them the meristem. And as mentioned previously, plant stem cells maintain their ability to differentiate all throughout their life cycle. So getting a meristem means you can actually then grow any part of a plant. So if you wanted to use it to produce flowers, you can use it to create rooting powder. They've got a huge ability, so just getting a part of a plant means you can then regrow that whole plant. Okay, so you could get asked to compare stem cells. As I said, there are three real types. So one, there are human embryo stem cells, which could become any cell or organ. The problem with human stem cells is we say they have a potential for life, which means they have ethical issues. Also, if you're using somebody else's embryonic stem cell to treat your disease, there is the risk of rejection. The second type of stem cell you would use would be an adult stem cell, which means you can only use it to treat some diseases, so it's less useful than an embryonic stem cell. However, there are less ethical issues, and because you'd be using your own adult stem cells, there is less rejection. And the final type, as I said, is a plant meristem, which can become any plant cell or organ, which are incredibly useful. They could be used to help with rare species of plants, so allowing us to bring back plants to prevent them from being extinct. They could also be used then to increase the growth of plants, so kind of cloning of plants, so cuttings, tissue cultures, and then it also allows you to create a large number of plants of desired characteristics, which we talk about a little bit more in terms of genetic engineering in paper 2. Common then 5-mark question you could get is going to be to evaluate human stem cells. I'm going to just set it up as a table. So first of all, what do we mean by the word evaluate? So evaluate are benefits and drawbacks. So So I'm going to split this into two sections. So first of all, I'm going to talk about the benefits of embryo stem cells and their drawbacks, and then we're going to talk about adult stem cells. So the benefits of embryo stem cells, already mentioned, is they can become any cell or organ, which means they can be used to treat diseases. Give an example. So you might be able to recreate a pancreas for somebody who's got diabetes. You might be able to treat paralysis, which is incredibly useful. So they can become any cell or organ. However, there are massive ethical issues with using embryo stem cells. They do have what we call the potential for life. So an embryonic stem cell could become anything, which is why some people think that we should not use them. The counter to that, though, and this is kind of, you can either get a benefit or a drawback. Every single year, there are lots and lots of wasted embryos which have been discarded as a result of not being used in IVF. So why not use those embryos, embryonic stem cells, as a benefit? And then, of course, you have adult stem cells, which have got a great use in terms of being used to treat blood diseases. But that comes then with the drawback, they can only be used to treat some diseases. So they're not as useful as an embryonic stem cell. There is a final type of stem cells that I'm just going to briefly talk about, which is something called therapeutic cloning. So that's where a stem cell or a cell is removed from an adult, and then so it's your adult cell, so it contains your genes, and then it becomes an embryonic stem cell, which means it can then again be used to create any cell or organ. However, this happens in a lab, and the huge risk of this is it often picks up viruses, which means then when you are using it on a patient or using it on yourself, there's a huge risk of that viral transfer, which could cause an illness with itself. So not only does it have them issues in terms of the treatment, it then has ethical issues as well, because you're using an embryo cell, which brings with the ethical issues of using the potential for life, and now there's some medical issues of the risk you could be transferring a virus. So commonly asked questions then that you could get in terms of mitosis and the cell cycle. So you need to match the stage then to what happens. We said in stage 1, the very first thing that happens in mitosis is the DNA gets copied. The nucleus then divides, and then finally, once the nucleus is divided, the cell divides in two. So the mass of DNA in a human body cell at the start of the cell cycle is 6 picograms. What is the mass of the new cells produced by cell division? Well, we said it earlier, mitosis produces genetically identical cells. So if it's 6 picograms then, it's going to be 6 picograms then. Okay, finally, a very common question that comes up is to look at the evaluating therapeutic cloning. Make sure you read the content. So, stem cells from human embryos can differentiate into any human cell. Research is being done into the use of embryonic stem cells in medical treatments. The long-term effects of using embryonic stem cells are not well understood. In therapeutic cloning, human embryos are produced using a donated human egg cell and a cell from the patient. The embryo produced contains the same genetic information. Stem cells are taken from the embryo and stimulated to divide to form the patient's knees. The embryo is then destroyed. So, what's the advantages? They give you some clues in there. So first of all, it has the same genetic information, which means there'll be no rejection. Second thing, then, it talks about the fact that they can be used to treat diseases. So treat diseases such as a new organ, e.g. a pancreas, or paralysis. So why would we not want to do it? Well, one, you're still using an embryo, which means there is a potential for life, which is our ethical issue, and there is a risk of virus transfer.